hits the retina, then sensory information more or less directly is related to a central region of the brain, the thalamus, and from the thalamus to the respective sensory areas. So in the visual system, it's the primary visual cortex. We will hear today about the primary somatosensory cortex, the area for tactile sensation, touch. Okay. And this kind of feed-forward process, bottom up, from the receptor to the thalamus to the sensory cortex, is then thought to somehow be coupled with additional information, which typically is thought to happen more in the frontal areas of the brain, higher order cortices, who knows what that means. And this coupling process is then thought to be the origin of perception. So you can rephrase this issue actually so to the question, how does the cortex transform sensation into perception? And that's kind of the basic question that my research group is interested in. But today I'm, of course, not going to reveal an answer to this question to you. We just, step by step try to disentangle how this may be working in the brain. So the question that I'm focusing on is essentially just the first part, and you will see how relevant this is already for this coupling idea later on, where the question is, how does the sensory cortex transform the input from the thalamus into some kind of output signal? So how is really the first stage of sensation happening mechanistically in the brain? And in contrast to these motivational pictures, we're not doing this on the human, but we're doing this on the rodent. You see here our beautiful model organism, the rat, also a bit the mouse. And we're using the whiskers, and you see the animal here having its whiskers. And these animals are really able to actively move their whiskers back and forth as an active tactile device, and they use this a lot. These are nocturnal animals, they live on the ground, so they don't see much under the ground, and thereby they rely pretty much on sniffing and whisking, a very coupled system, and can touch, perceive objects, shapes of objects, other mice and rats, and so forth. So it's a very important model system. So the question that we're investigating, how is input from the whiskers transformed into a percept, for example, of an object or a shape? Just for those who have never been exposed to the whisker system, why is it a good model system to study that? Well, sure, it's a mammal, but why the whisker system? Why not the visual system or the auditory system? Well, the whisker system has a few advantages for this task. One is that it's been known for a very, very long time that these animals can do relatively complex behaviors with sensory input from just a single facial whisker. So you see one of these classical experiments from the 1980s here, so-called gap crossing experiment. Animal comes in darkness to a gap, tries to reach over to the other side, and then, depending on the gap size, decides whether to jump or not. And this input, as you see, this is the top view here that we did onto this gap. All the whiskers have been trimmed in this animal, and it's only moving one of its whiskers, and it's able to make this decision reliably with just sensory input from this whisker. So we can actually reduce the problem of sensation to just input from a single whisker, which makes things a bit more easy in our case. So you can ask, how is the input from a single facial whisker transformed into a percept? The other advantage of the whisker system is that it's highly somatotopically organized, which means there are segregated areas in the brain, stem, thalamus, and cortex, which are specifically responding to the input from that particular whisker on the snout. So in other words, you have an anatomical reference frame, and you will see a few pictures of that, which allows you to do experiments over experiments with different tools, different modalities, different questions on the very same structure over and over again, which is a good advantage of the whisker system. So this nice somatotopic organization and the single whisker-based behavior capability makes this maybe a tractable system for us to study sensation and perception. How do we do that? Well, I guess in this workshop, it's not a surprise that we believe modeling is an important part of this kind of equation. And the idea that we have is that we would like to essentially build an extremely detailed model of the cells and circuits involved in whisker information processing, and to use these kind of data to generate multi-scale models that allow us to, as closely as possible, mimic in silico the kind of in vivo experiments we're doing. So the idea would be to measure, and you will see a few examples today, to measure precisely the activity of neurons in the living animal during sensation or perception or some kind of behavior, and to then use these data to combine it with networks models and cellular models to then ask what of these very complex but realistic models could reproduce our in vivo observations. Why do they reproduce these in vivo observations? And what would they predict are the mechanisms that could account for our observations so that we can go back to the experiment and test by doing these manipulations which we previously did in the computer to test whether these manipulations really yield the kind of outcome that we would expect if we have understood something, and then we step by step iterate to get a more detailed answer to our questions. So how do we do that? Well, I will start with how do we reconstruct the connectivity? And we heard a lot about connectivity and definitions of connectivity in the last few days. So when I talk about connectivity, I mean the synaptic connectivity. Synapses formed between neurons. Where are the synapses? How strong are the synapses? Where do these synapses come from? What are the pre- and postsynaptic cell types, etc.? And to do this at a dense level, dense meaning we try to account for all the synaptic connections of all the neurons in a particular piece of tissue. So the idea is to generate dense network models to constrain our simulations with the empirical observations made by different connectivity measurement approaches. So one of the very basic questions we started with back in the days was the simple question, so how many neurons are we talking about? How many neurons are involved in processing the information from a whisker, from a different whisker, and so forth? And of course, if you look into the textbook, you find about cortical columns, about 10,000 neurons per column or something, but that is a wild guess, right? That is not a measurement. So we therefore decided to really essentially do the simple, yet it turned out relatively difficult task to just count the number of neurons in the somatosensory cortex that is associated with the vibrissae, the so-called barrel cortex. You see a cross-section through the brain here. If you squint your eyes, maybe you, that's how the frog also sees the barrel cortex here, which is this more dense region over here. You will soon see better. These dark spots are the blood vessels, so it's sliced tangentially, so the blood vessels are running perpendicularly. So when you zoom in, you see the cell bodies of all the neurons, and then even without machine learning at the time, we were able to detect the soma locations of these neurons. And then if you counterstain these sections with so-called GUT67, which is a marker for inhibitory neurons, you actually, for whatever reason unknown, actually see the somatotopic organization of the vibrissae, where each of these columnar shapes here, the cylinders, represents one of the vibrissae, and neighboring columns represent neighboring vibrissae on the snout. This would be the vibrissae at the bottom of the snout, the vibrissae at the top of the snout, front to back. So we could essentially thereby count what are the number of excitatory and inhibitory neurons associated with each individual whisker, which led to some very surprising and interesting insights on the overall organization of the number of neurons, where we essentially found, yes, on average, is about 10,000 neurons for such a column. However, the whiskers representing those at the top of the snout have only about 9,000, and they're progressively increasing all the way to here, where these whiskers have about 30,000 neurons responsible for their processing. So that's already an important information when you try to understand how these neurons are processing. We did the same.
So when you do the, when you do the single whisker thing, do you try with different ones of these?
Exactly. You can. There are, of course, question, and we will come to this in a second, how we look at receptor fields for different whiskers and how these receptor fields are related and so forth. What's the organization also of multi-vector inputs, not single whiskers per se, but multiple single whiskers at the same time, etc. So you can do a lot of interesting things. Also, when the animal is actively moving its whiskers to do stuff, I will show a few examples of that. The different colors represent essentially the rows of the vibrissae. Red meaning the vibrissae at the top of the snout, and then there are 5 rows: A, B, C, D, E, and blue would then mean those at the bottom of the snout. And since I told you the somatotopic organization is not restricted to the sensory cortex, it's already happening at the brainstem and the thalamus, you could actually then do the very same in the thalamus. So you see the somatotopic organization of the vibrissae in the thalamus, and then count what is the number of input neurons to one of these columnar units. And we found the very same relationship, that it also goes from low numbers to high numbers. where you have about 100 neurons in the thalamus responsible providing input to 10,000 neurons, and about 300 neurons to 30,000 neurons for those at the bottom. So we have at least an idea how many neurons are we talking about, how many neurons provide the input, how many neurons could potentially receive the input from those neurons in the thalamus. But the parts list is, of course, not just excitatory and inhibitory neurons and their numbers, but what are those neurons, essentially? And this has been a painstaking process to essentially, like, doing stamp collecting, doing many experiments and probe what is the structure and function of the individual neurons, what kind of neurons do we actually find in the thalamus and the cortex of the system, and how could we categorize them, at least roughly, to know what are the basic and primary cell types involved. And you see here the result of this, which I believe is now more or less the accepted view of the primary cell types of excitatory neurons in the rodent sensory areas, where you see already an enormous diversity across the different layers. So the pia surface would be down here, and the white matter would be here. This is the thalamocortical axon that provides the input, and these are intracortical neurons. In black is always the dendrites, and the colors represent the axons of the respective cell types. And you can already appreciate the enormous complexity of these individual neurons. These are in vivo labeled neurons, so you see the full axonal extent of these neurons. And it turns out that it was much more elaborate than we initially thought. Elaborate meaning such a cell, for example, this guy here, and you will hear a bit about it, so-called layer 6 cortical neurons. You see its axon here in light red or whatever that color is. Such a neuron has on the order of about 25 centimeters of axon just locally in the somatosensory cortex. And so it's enormous. And you see its massive extent here, where you see already the big implications that, yes, the input is relatively specific for whisker, but these neurons immediately spread the information throughout the whole sensory area in various different ways, in a layer and cell type specific manner. So this kind of idea that one of these column processes information from just one of these whiskers is already kind of seems invalid when you just look at the axons of the receiving cells, which immediately crosstalk across the whole cortical area. And that was a surprise at the time. We also did, without going into details, to do this for the inhibitory cells, which is an even more challenging task, because there, even though there's just about 12% or so of the neurons in the cortex are inhibitory cells, the diversity of their morphologies, molecular identities, or electrophysiological properties is much more enormous than those of the excitatory cells. And it's still challenging to find some consensus to how to parcellate inhibitory neurons into some meaningful groupings. And here's an attempt that we currently did, which I don't want to go into detail, but interestingly enough, we found a very nice relationship that how, even though you see an enormous diversity, for example, in the group of fovalbumin and somatostatin expressing cell across the layers, you can still separate them based on the very same morphoelectrical criteria across different layers, which seems to be a very interesting and promising avenue. So having now essentially this parts list, what we did is to use this data that we have systematically acquired over almost 2 decades to build a dense model of the somatosensory cortex. Dense model meaning that we account for all the neurons in this area, for the amount and distributions of dendrites and axons, for the spines along the dendrites, as well as for the boutons along the axons, to get essentially a dense model of the neuropil structure. Dense model of the neuropil structure meaning there's no connectivity in this model. This model is purely distribution of dendrites, somata and axons, spines and boutons, the raw material, so to speak, for connectivity. And we made some predictions from that. If you would chunk up in an electron microscope and densely look how many meters of path lengths would you find in such a volume for dendrites, axons, how many branches, how many snippets, how many synapses, etc., and compare this to the volumes that were accessible at the time with dense electron microscopy reconstructions, we found it's a relatively close match between what is expected and what is found. And just to make clear the complexity here, this is a 50 by 100 micron small volume, and you already find about 3 meters of path lengths in this volume, which represents about 40,000 something branches and 500,000 synapses in this volume. Right? So just imagine the volume of the barrel cortex here is about 6 by 5 by 2 millimeters. So it's an enormous tissue with an enormous number of dendrites and axons. About 500,000 cells, about 5 billion synapses in this cortical area. So having now this anatomical model of the neuropil structure, which we could show is robust given the kind of data that we have collected. So in other words, collecting more and more anatomical data would not change the predictions of the neuropil structure for this particular volume. We asked, how could we then infer connectivity from such a structural model? And of course, this is an enormous complex task because we know that synapse formation itself is an extremely complex process. And synapses form at the molecular level based on many, many conditions. Some synapses form, for example, depending on the activity of the pre- and postsynaptic neurons. Other synapses may form on the specific genetic identity of that cell, the cell type identity, the particular location of a synapse, whether this neuron is already connected to another neuron of a particular type, and so forth. And all these mechanisms have been observed at the molecular level, and therefore it's, of course, an extremely challenging task to infer connectivity in a meaningful way. So the idea that we proposed a few years ago is to essentially formulate such synapse formation strategies that are observed at the molecular level in terms of assumptions, where the simple idea is that you formulate a kind of a preference to connect to a particular postsynaptic neuron with a positive number and a negative preference, so to speak, with a negative number. And if this parameter would be 0, essentially the cell has no preference to connect to anyone. So it's kind of a random synapse formation process, so to speak. And these parameters cannot be conditional on the cell type. I'm an excitatory neuron, and I only like excitatory neurons to connect with. I only like inhibitory neurons. I like particular subtype on a layer or a subtype of an inhibitory neuron. I must be correlated, have my activity correlated, and then I form a synapse, or I only form synapses on spines, on stomata, on axon initial segments, or on dendritic shaft, etc. So you can make any kind of assumptions that you want and test what would be the predictions of those assumptions. And by meaning any, we developed an inference approach together with people in Tübingen. It's called a simulation-based Bayesian inference approach that allows us to find all of these parameter configurations that are consistent with the empirical data that we know. And with this kind of inference loop, you can then identify what kind of assumptions are necessary, what kind of assumptions are sufficient, and maybe what assumptions are best suited to, for example, explain why in electron microscopy, often you find these clusters of synapses between neurons, where one particular neuron forms more than one synapse on a particular piece of dendrite with some other neurons, which is referred to as synaptic clustering. You can also look at how cell type and location-specific connectivity could arise from such constraints. For example, we see here between some layer 5 cells that the connection probability between them decays with intersomatic distance. Or what we heard a bit about the other day, higher order connectivity motifs, which generally are found to deviate from those expected in a random network. Why are these neurons, for example, form a fully recurrent loop? And what kind of assumptions about synapse formation strategies would be needed to account for such observations? So with this approach, you can thereby identify and constrain your models and find different variants of assumptions that could account equally well, in many cases, for all of these empirical observations. So thereby the idea is to then take our in vivo recorded neurons for which we know the activity, for example, in response to different vibrissa, and embed them at their position in the somatosensory cortex, and then generate millions of synaptic input patterns to such an in vivo recorded neuron. All of these connectivity patterns from the network are consistent with what we know from all kinds of measurements of many, many decades by many, many people with different approaches. But where we can thereby test to what degree the simulations which we later on do with these connectivity constraints, which of these simulations actually survive, which of these simulations are robust with respect to variations in the morphology of the neuron, the number, locations, distribution, strength of synapses, and even with respect to different assumptions that we created in the first place to generate these models. As a little outlook, of course, now it's very tempting, and with the rise of dense EM data becoming more and more accessible at larger volumes, to directly use these kind of inference approaches on dense electron microscopy, not taking the detour that we did of kind of building a dense model, but using the dense data directly to constrain the connectivity models. And you see here one of the data sets from Jeff Lichtman's group, where they looked at the human temporal cortex, where the synapses are really defined based on measurement of a postsynaptic density between a spine and a bouton in this case, where we do exactly the same approach, where we know the connectome at the nanoscale and can thereby, for each of these connections and possible connections, infer what assumptions are necessary that could have generated this connectome from this anatomical data. And just as a little teaser and outlook, we did this now for a data set from the mouse visual cortex from the Allen Institute and this human data set, where you see here just some pairwise statistics for simplicity, the number of synapses between excitatory, inhibitory, excitatory, and so forth, the number of excitatory to excitatory somata, dendrites, exon initial segments, and so forth. For these two data sets, in black is the empirical observations. In gray, very interestingly, would be the predictions if we forget about synaptic selectivity mechanisms. So all these alphas are 0. And you see, it's not so bad. And we have published this a few years ago, how the impact of the anatomy itself leads to these highly specific and non-random wiring patterns, but it's not perfect, right? So there are deviations, of course, from the anatomy itself, where then the inference using synaptic selectivity mechanisms can really perfectly approximate and capture what has been empirically observed in both of these data sets. And then you can start looking, what are the predictions of attraction and repulsion mechanisms needed to explain those? And I've just found it interesting, I have no clue yet, this is very new, that the assumptions that you need for both of these data sets, even though one is in layer 2, 3 of the mouse visual cortex, one is in layer 5 of the human temporal cortex, very different connectivity patterns. But the assumptions that you need to explain both of these observations are very, very similar between them, which I found remarkable. And in particular, that there's predicted a strong kind of attractive mechanisms from inhibitory neurons to excitatory neurons, whereas you have a more repulsion needed to account for excitatory to excitatory connectivities. In other words, you would see much more excitatory connections than or you would expect to see more than you actually see. And for the inhibitory neurons, it's the other way around in both of these data sets. Okay, so just, yes.
Sorry, in this, did we account for the big difference in the ratio between excitatory and inhibitory neurons between human and the rodent?
Yes, this is agnostic in many cases of the numbers and so forth. It really takes the whole data set, right? It takes the whole data set and looks for each synapse. So essentially for each, not even that, for each presynaptic structure and each postsynaptic structure, it calculates the probability of forming connection with all the possible connections there are, whether it's an excitatory or inhibitory. You can make this assumption without excitatory inhibitory and just look for that and be completely agnostic. So it's automatically normalized, if you wish, to the overall number in the data set. You see here 130 million synapses in this data set, and this data set is much smaller. It's only about 3 million synapses in the data set. So you get a number for each synapse of each neuron. Right? So it's not a matter of how many, right? It's only a matter who could I potentially connect to, right? So the inferred specificity matrix has the same resolution as the wiring diagram. Okay, so this is an anatomically detailed model. So this tells us a bit who could be talking to each other and how and where. But of course, this is only part of the equation. First, how a neuron takes this input, and we heard a bit about it yesterday, how these neurons take such an input to transform it into an action potential output is an at least equally challenging question to be addressed. And therefore, we are generating functionally realistic single neuron models that capture the intrinsic physiology of these neurons. And I want to focus on one of the cell types, which is one of our preferred cell types, the so-called layer 5 terminal tract neurons in the barrel cortex, because there has been increasing evidence from more and more labs, in particular from Matthew Larkham, that the responses of these neurons in the primary sensory area of the whisker system are directly linked to perception. And I will come to this in a second. And these cells are interesting in a way because, so on the one hand side, these are the major output cell types of the cerebral cortex. So those are the neurons which send axons to subcortical structures. So everything that leaves the cortex has to go through these neurons. So therefore, they are implicit or explicitly of behavioral relevance. So without them doing something, nothing will happen. Everything stays in cortex. And the other thing is that they have enormous and large dendrites, which essentially span across all layers of the area. So they are a major integrator cell type. They receive on the order of about 30 to 35,000 synaptic inputs per cell from all local populations, but also from many thalamocortical and cortical pathways. And thereby they are thought to be really critical for transforming any kind of input that these neurons receive into output. And what has been interesting is that these neurons can generate, and I will come to this in a second, bursts of action potentials upon sensation, and that these bursts of action potentials seem to be necessary for perception, at least in this case of whisker stimuli. So in other words, if you manipulate the channels of these cells, in this case, the calcium channels and the distal dendrites, these neurons would not respond with bursts anymore to the sensory stimulus, which then lowers or raises the threshold for perception. So you can actually tune by modulating the calcium domain in these neurons, how strong is the threshold for perception that you need? How strong do you need to give a whisker input here? The origin of this is, however, unclear. But this is one of the motivation to build biophysically detailed models for these kind of cells to be able to understand how they take such an input and transform it into an output. This has been very challenging. The physiology of these neurons has been studied for decades. It's probably one of the most and best studied cell types in the cerebral cortex at all. Yet, despite the knowledge of so many channels that are expressed in these neurons, so many physiological features, the ability to capture this in a single neuron model has been a major challenge for the past. And the major challenge, there are many challenges, but the primary challenge seemed to be to account for the ability of these neurons to generate calcium spikes, calcium action potentials in their dendrites, and to model the ability that these calcium action potentials in the dendrites can modulate the somatic response. So this kind of crosstalk between dendrites and axons with active mechanisms is extremely difficult to capture in a model. And I'm just illustrating here one of the experiments, which was kind of key and led to this observation, is where in slices at the time, you would record at the soma and inject a little current at the soma, you elicit an action potential, so no surprise here. But then if you record with a second pipette in the dendrite, you see that this action potential actively backpropagates into the dendrite. And if you would then give a tiny current up here that coincides with this backpropagating action potential, you would suddenly elicit a very strong calcium spike, calcium action potential in the dendrite, and the same input at the soma that previously generated a single action potential would generate a burst of action potential at the soma. So you can thereby, this mechanism is called coincidence detection. Essentially, one input to the soma causes an action potential, one input to the distal dendrite causes nothing, but if you do both at the same time, 1 plus 0 is 3 in this case, right? So this is a non-linear mechanism. Okay, and this has been extremely challenging to capture in models. As I told you, there's been a lot of people that collected this kind of data. You see here from many decades from our collaborators, but also from previous studies, how you measure at different dendritic and somatic regions very precisely the shapes of action potentials, the interspike intervals between different bursts, the after hyperpolarization we heard a bit about yesterday, and so forth. And you see there's variability, of course, from cell to cell, from trial to trial, which relates a bit to the morphology sometimes, sometimes not. And these models are supposed to capture all of these features. And I'm just showing about, 10 features here. There are many, many more for different kinds of somatic and dendritic current injections, which we're trying to capture from time-varying signals at the dendrite and soma, resonance frequencies, and so forth. And the long story short is that we are now able to do that. And I'm not going to go into details how we accomplish that. But what you see here is essentially the parameters of channels that we know are expressed in these neurons in different somatic, dendritic, and axonal domains, different potassium, different calcium, different sodium channels, different intercellular mechanisms, and so forth. And then we can now explore this entire space of possibilities and ask, okay, which of these models capture the intrinsic physiology that we see in these neurons? And you see here, the intrinsic physiology in the cell doesn't change. The expression of channels changes a lot. And even the way how these channels are mechanistically used to do the very same physiology. So what we observe here is an enormous degree of freedom, so to speak, in how channels could be in principle expressed, how these channels could be combined to then explain these many, I think, 80 different physiological features that we have now used to constrain these kind of models. And this is not an effect of the morphology. So you see for one particular neuron here, you find very different ways how the very same morphology can implement its function. Here's a very different morphology. You find the same diversity of these kind of models. So essentially the idea is here similar to the connectivity approach that for each of our in vivo recorded neurons, we can generate millions of such biophysically detailed models, all of which capture what we know about the intrinsic physiology. But we can ask again, which of our simulations later on are robust and survive with respect to these variations in channel distributions, channel utilizations, or morphological features. Okay. Of course, that's quite an effort to do that. And the question is it worth it? Right? Does it help us to answer some of our questions, what these neurons do in vivo? Does it help? Does all this detail matter? And one of the questions that we wanted to address, of course, is what I mentioned in the beginning. How is input from a thalamus transformed by the somatosensory cortex in some kind of output signal? And there we went to what is known, of course, right? And this is the kind of what is called the canonical cortical circuit idea, which has been proposed by Kevin Martin and Rodley Douglas for a long time. It goes back to the late 70s on research in the primary visual cortex of the cat. It's been shown in the mouse, etc., in the whisker system, auditory system, where the idea is that there's kind of a serial flow of information where the thalamus first provides input to neurons in layer 4. These neurons in layer 4 then relay this input to the superficial layers 2, 3, and these layer 2, 3 neurons would then drive these layer 5 neurons that I was talking about, and these layer 5 neurons would generate an output. This is the canonical cortical circuit idea, and there's quite some evidence in terms of anatomy at least, but also physiology, that this is true. However, it's been known for a very long time as well, and René Bruno here showed this very beautifully for the Vibrissa system, that there is some inconsistency with this picture, where if you record from neurons across the cortical depth and measure how fast do they respond to the sensory stimuli, you find that the neurons in layer 5 respond before the neurons respond in layer 4. So how can it be that the output neurons respond before the input neurons? And he could even show, we take this out, we manipulate it, and this has no impact on the sensory evoked responses of the layer 5 neurons. So he thereby concluded a decade ago, well, it seems the canonical circuit is all fine, but it's not driving the cortical output. Maybe the thalamus could directly drive the cortical output. So he would propose such a bimodal input pattern, where layer 4 is driven by the thalamus and the same thalamus drives the layer 5 cells directly. However, as always, there's also, if you do another experiment, it's conflicting with that idea as well, where many people have shown that when you record in the thalamus, how do neurons respond to the different whiskers? And you see here this for 9 different whiskers tested, that in the thalamus, these neurons like their somatotopically aligned whiskers. So they respond to the somatotopically aligned one. If you do the same experiment with one of these layer 5 cells, they don't care. They respond to all whiskers with the same latency. So how can it be that this is driving that, given that the axons are not going there? So this neuron over here doesn't see that input. So how is it responding? At the same time. And what's even shown directly the opposite, that the input from the thalamus is not sufficient to do that in contrast to that. That's physiology. So could our modeling detailed stuff help us to answer this question? What is the origin of these fast onset and the broad tuning of these neurons? What we did is we measured what are all the presynaptic populations doing. How are layer 2, 3 neurons, the different excitatory cell types in layer 2, 3, layer 4, layer 5, and layer 6, the different inhibitory cell types, the neurons in thalamus, how do they respond? to essentially activate our network models consistent with the recording data that we have from these different populations, to then make predictions. What would we see in a layer 5 cell? How would they respond? And to cut a long story short, we were able to capture both of these phenomena. So these simulations predicted without any tuning, right? It's just taking what we have, that yes, these neurons would respond equally fast to a broad range of whisker stimuli, and they have even the variability that we see in vivo of the receptive field shapes and trial-to-trial variability is well accounted for by these models, which allowed us to ask why. Why would the models capture this phenomenon, which we didn't tell it to do? And the answer was this: these layer 6 cells, which I briefly mentioned in the beginning, where we found that these layer 6 cells, you see an example here in red, is not a layer 6 cell per se. So these neurons, you see here the thalamocortical axon with dense innervation of layer 4, but the very same thalamocortical axons have also a second innervation peak at the border between layer 5 and 6. And similar to how the layer 4 cells cluster here around these innervation domains, these layer 6 cells cluster around the border between layer 5 and 6. So when you look at the distribution of axons, thalamocortical axons, this is where the layer 4 cells are located. These are where the layer 5 cells, layer 6 corticothalamic cells, and these cells sit right in between, where the axons are innervated. So essentially you have the same axon has two innervation peaks, and there are two excitatory populations clustering around it, one in layer 4, which does this vertical flow of information in the canonical circuit idea, and one that does a horizontal spread of information in the deep layers. And essentially every time a sensory stimulus comes in, these neurons are reliably activated, relay the information immediately across the whole cortical area to drive these layer 5 cells, and thereby give rise to these fast onsets and broad receptive fields, even before layer 4 knows there was an input. So essentially, the canonical circuit idea we could augment with the idea that there is a second population that is driven by the same thalamocortical input and drive these layer 5 cells to generate these fast and broad responses. And we could also show by manipulations in vivo that by depriving these cells from this input, you can actually abolish the sensory evoked responses completely and even delete a single whisker out of the receptive field of a single neuron. This also explained why Randy's manipulations up here had no impact on these fast responses, because they're driven by this deep layer pathway. However, the question that remained is, why is the thalamus providing direct input to these neurons, which Randy, Bruno showed very nicely physiologically. Other people, Svoboda and friends, and many people showed that there are connections directly strong from the thalamus to these output neurons. But we showed both in simulations as well as in vivo that this direct sensory input to the output neurons is not necessary to drive the sensory responses. Essentially, you take it off, they still respond. So it's really these layer 6 cells that do that. So why would these neurons receive direct sensory input if it's not driving any response? And an idea came from a very recent observation that we did where we showed that this layer 6 pathway is necessary to explain the responses of the neurons per se, but it cannot explain burst responses in these neurons. So you see here, as soon as you start deviating on a trial, but on a single cell level and get more than one extra potential elicited in a response, these pathways fail to predict the generation of these kind of burst responses. So it seems these layer 6 pathways are necessary to drive the response, but are insufficient to account for the burstiness that occurs sometimes in these responses. And we thought maybe the thalamus has something to do with that. Maybe the thalamus is needed to modulate these responses, its own responses with bursts. And as you remember, there is this evidence that these bursts, in particular these bursts, may be important for this transformation of sensation into perception. So we looked into this without going much into details. What we first did, we wanted to precisely measure where are really the thalamocortical synapses on those neurons when we simulate them. So we did an approach where when we recorded from our neurons, we would post hoc then reconstruct these neurons and really with super resolution microscopy identify every primary thalamocortical synapse on the dendrites of our in vivo recorded neurons And you see one example here where the green dots represent the thalamocortical synapses. And when we converted this into a density plot, we made a very surprising observation. Where you see here, and maybe you remember that this bifurcation area of the apical dendrite is the region where the calcium spikes are initiated in these cells, where the calcium channel density is very high. And we saw that the thalamocortical synapses seem to cluster exactly there, that they have the highest density specifically around the bifurcation point where the calcium channels in these cells are located. And this was an observation that we had no matter which neuron we looked at, no matter where the calcium domain in this neuron was, whether it's deep almost in layer 5, in layer 4, or all the way up in layer 2. The thalamus seems to know where the calcium channels of the cell are. Or maybe the other way around, the calcium channels get implemented where the thalamocortical axon hits the strongest. No matter what, this seems to indicate, well, if sensory input comes in, it provides some kind of strong input to the calcium domain. Maybe this could generate these bursts and the coupling mechanisms that we saw before. So we did the recordings here, in this case, passively stimulated all the whiskers. And what you typically see is either the same neuron from one trial responds with a single exponential. In the next trial, it may respond with a burst of 2 exponentials. In another trial, with a burst of 3 exponentials. And for the very same cells, we optogenetically activated these thalamocortical synapses, for which we had recorded. And this gave us an interesting observation that by direct activation of the thalamocortical synapses, we could drive a single exponential or a burst with 2, but never a burst with 3 exponential, indicating, well, maybe even the number of exponentials within a burst reflects different mechanisms, which was even further supported when we repeated our experiments in animals that actively move whiskers to touch an object in this case. We found, yes, we see the same that we saw here for multi-whisker deflection in an anesthetized animal. We see the same in awake, 1, 2, or 3. We even see that the overall response probability of these cells is identical under these very different experimental conditions. The occurrence of these 2 bursts of exponentials is not different between these very different experimental conditions, but these bursts with 3 exponentials occur much more frequently in an awake, actively sensing animal than in a passive situation in an anesthetized animal, providing again evidence that these bursts with 3 exponentials may have a different origin than those with 2 exponentials. That's a beautiful scenario for a simulation to test. So we essentially did the same that we did for the receptive field story, recorded from all the possible presynaptic populations what they do under these different experimental conditions to activate our network models. And then again, these simulations, or about 10% of the possible simulations or the possible models, predicted exactly what we observed. Sometimes you see a single, sometimes you see a doublet spike, sometimes you see one of these triplet bursts. And we could thereby ask in these models, what is needed to explain them? And as no surprise, for a single exponential, we found what we had reported before. The layer 6 pathway that I alluded to is sufficient to explain why the neurons respond in the first place. And you don't need anything from the thalamus or active dendrites to explain any of it. For a doublet burst, it turns out, yes, the input from the thalamus is necessary. So if you would take away the input from the thalamus, there is no burst at all. But this has nothing to do with active dendrites, nothing to do with these inputs to the calcium domain. This is purely strong input to the basal dendrites. In contrast, for these triplet bursts, we needed all of that. No model without this could explain these bursts with 3 exponentials. And what we even found is that this generation of calcium exponentials is conditional on the amount of pre-stimulus inputs that impinge onto the distal dendrites. So in other words, the prediction here was very clear. The reason why in one trial you see a triplet is there was more input to the distal dendrite at this time to this neuron than in another trial, and thereby the same input due to pre-stimulus inputs from who knows what got modulated, and the thalamus was able to drive one of these calcium spikes and modulate its own input with these bursts. And the concrete prediction from these simulations essentially is that the probability that this happens should essentially increase the more inputs come in here before a stimulus. And this is another experiment which we then did to test that, which is now again the in vivo situation where we optogenetically activated the thalamocortical synapses and monitor how strong is the activity intracortically just in the 10 milliseconds before we turn on the light. And you see here at the single cell level, the probability to observe one of these triplets versus one of these single action potential responses essentially flips the more intracortical input is provided before we turn on the light. So to sum this up, what do we learn about sensory cortex and its transformation from thalamocortical input? Well, we found that the direct sensory input via the layer 6 cells, but also the direct input of the basal dendrites from the thalamus is driving the responses, the sensory responses in these major cortical output cells. So it's the feed-forward drive from the thalamus with the help of layer 6 cells that accounts for the very fast and broad responses that these neurons have. It's not the canonical circuit. However, what we could now show is that the non-sensory input preceding the stimulus is determining what kind of response occurs in these cells. So essentially, the message is here that already the very first action potential that leaves the cerebral cortex, even before layer 4 knows what's going on, are modulated by the ongoing activity in the cortex at the time the sensory stimulus happened. So this idea that the sensory areas process sensory information and then relay it to some higher order area where it's then coupled with something is probably true. But the other way around seems also to be the case, that all these areas which are active before the stimulus even happens modulate already what the first sensory evoked responses are doing, which of course has a huge impact. Which now sets the stage to investigate more closely, why are these bursts linked to perception? Which we have no clue why, but we at least have now an idea what are the origins of these kind of burst responses, and thereby we may have a handle to better understand why these burst responses that these neurons elicit are linked to perception. And in the last minute, maybe to link it more to the kind of research that most of you guys do here. This is a wild guess, so don't take it too seriously, right? But one thing that I find intriguing is to think about it like this. So these would be the layer 5 cells, and one of their primary targets is the higher order thalamus. So these layer 5 cells project to the higher order thalamus. It's one of the primary pathways. And these neurons in the higher order thalamus provide feedback to the same cortex, but also to many other cortical areas, including the frontal cortices. And these neurons form giant synapses in the higher order thalamus, very strong giant terminals. So in other words, action potential is elicited, comes into one of these thalamic neurons, they like it, they have very strong synapses, they spike. Looks a bit like this. Action potentials come in. However, these synapses are very strong, but they're also strongly depressing synapses, which means there are essentially low-pass filters. If bursts of action potentials arrive here, they're essentially filtered out. A single burst coming in on some branch of this neuron will not result in a stronger response than having no burst. However, if multiple bursts, and this has been shown both in slides as well as in vivo, if multiple bursts arrive synchronously on different parts of the dendrites of these neurons in the thalamus, these neurons can overcome the depression of the individual synapses and start to facilitate, and thereby generate bursts of action potential themselves, have increased input back to the cortex, and thereby increase further downstream processing in the cortex, which has been demonstrated by Alex Groh and some other people. So it seems intriguing to think a bit like this, that what this coupling mechanism does is essentially to allow all the layer 5 cells triggered by the input, which is essentially a time-locked signal, to simultaneously increase the probability to generate bursts, and the ability of these bursts could thereby facilitate the activation in thalamus and subsequently in cortex. And this is reminiscent, at least to some degree, to what has been observed in the humans, where if you do these kind of experiments, in this case for a visual task, and ask the participant, do you perceive the image or not, that in cases where the percept hasn't been established, it seems that the activity essentially gets stuck in the primary visual area. It's not able to activate a global kind of processing stream. In this other scenario, where the same stimulus by the same patient or participant, not patient, is perceived, you see this reverberating activity somehow gets activated. So something in the primary sensory area seems to trigger this global ignition. And that's actually one of these hypotheses, the global ignition idea, and you probably know better about it than me. And Matthew Larkham has kind of tried to relate this to these layer 5 circuits, where it's one of the possibility that these are the neurons that are trying to communicate this by their ability to change their activity to burstiness, facilitating transthalamic communication, and thereby cortical-cortical communication in a large scale. So it may be the reason why these bursts in the primary sensory area are strongly linked to perception could be the case. Well, they are maybe the ignition point. This switch, depending on your pre-stimulus activity, what the sensory stimulus does, has a huge impact. Yes, it's just two more action potentials, but it can have a huge impact downstream and thereby on cortical processing, and thereby lead to a completely different global dynamics than what you would have without having the proper pre-stimulus information happening. So with this, maybe coming back to the initial idea, you should still see the frog. I have changed your brain now for the rest of your life. You can't get this out again. We have still no clue, of course, how all this works. But what we demonstrate here is that the thalamus itself, so the first stage of sensory processing, triggers the coupling of sensory information, its own sensory information, with pre-stimulus inputs, and that these pre-stimulus inputs may or may not, who knows, let's see, lead to a change in global dynamics. But what we have is a new physiological signature now of this process, these bursts with 3 action potentials, so we have an idea what to look for, how to test it, and how to see which of these different pathways could generate such signals and what happens downstream. Maybe get a bit closer to this idea, how your expectation may change what you see and perceive. With this, I think, of course, the people that have contributed to this and the funding agencies, and yeah, I'm done.
Thank you, Marcel, for that interesting talk. We have questions. Yes, Dr.
Thank you. I think it's amazing that you can get like this model with like all these neuronal types and everything. And of course, my question is like, I know it would be a tremendous amount of supplementary work, but how do you think not accounting for glia? That's what you're especially, I mean, because you look at this special burst and you see that this calcium input is very important, and of course, like. Well, astrocyte, for example, or about calcium regulation, and things so.
Yeah, there's a lot of things we ignore here, right? And neuromodulation in particular is one of the candidates which has definitely a strong impact on and neuromodulation and neuroglia are, operating and interconnected at the same time scale. And many of these receptors, and we do this in collaboration with Dirk Feldmeier actually in Jülich, looking at how are different neuromodulators expressed on different connection types and so forth, and what could their impact be, with another collaboration with Mickey London at the Hebrew University, who's looking into this. And I think these slower time scale changes, where the microglia may be involved in releasing certain transmitters or facilitating release of certain transmitters in neuromodulator pathways, this is a huge way how you can change the system. And we see a lot of evidence for specificity of receptor expression, also depending on the pathway, in particular in these layer 6 cells, where these layer 6 cells are under strong neuromodulatory control, which kind of makes sense, where you can essentially tune how strong does the sensory versus the non-sensory input do something, depending on some condition, right? Yet, that's already complicated, right? And I don't want to make it more complicated than it is right now, but this is definitely something to be considered, yet we ignore it for now.
Thank you for the great talk. So you showed that you need this pre-stimulus high activity to generate the calcium spike to get the 3 spike burst. And you said you measured the pre-stimulus activity. How did you make sure that this actually arrives at the distal dendrite, and what did you explicitly measure?
Exactly. This is something we cannot do yet, right? And it turns out, and the reason we cannot do that, because it's exactly the opposite of what you said. It's not some strong input, right? It's not a very, what we show is you don't need a very precise time-locked strong input to the distal dendrite. So essentially, it's the overall state of the distal dendrites that is influenced by a longer period of time, up to what we estimate about 40 milliseconds. And in other words, it makes a huge difference whether the calcium domain is at minus 55 or minus 54 millivolts at the time when the thalamus hits. Okay, and this 1 millivolt difference is about what we estimate about 2 or 3 synapses per millisecond difference in the distal dendrites. So we cannot do that. So we neither have the resolution nor the precision to measure that directly in the distal dendrite. What we, however, show is via simulations in this case, what would you expect if you make the input stronger in the basal dendrites? What would you expect if you make it on the dendritic trunk? What would happen in the distal tufts? And what is the most effective way to elicit this coupling mechanism? And you actually can show directly and provide evidence why that inputs somewhere else have no impact on that. So they would increase, for example, these doublet bursts or maybe increase response probability per se, but not these triplets. So in these experiments, we can essentially just measure the global activity increase in cortex, right? Not where, but we show it's everywhere, including the superficial layers, right? And see this phenomenon that this change of pre-stimulus activity in the sensory cortex has essentially only an impact on these triplets, and that in the baseline condition where there is no increased activity, you never see a triplet. This was this experiment I showed. You activate the thalamus, and it's the same neuron. You record, shine a light on the thalamocortical synapses, never a triplet. You get more and more increased cortical activity, and the same cell triplets. And the more intracortical activity is, the more triplets originate. So it's direct, indirect evidence, right? But the closest you can get so far with experimental techniques to try to measure this phenomenon.
So there's no cell type in which you could isolate, which primarily only targets the distal dendrites.
Everything targets the distal dendrites. So many local pathways target distal dendrites, the higher order thalamus, and increase shining light on something leads to a lot of things. Yeah, so yeah, believe me, I have wrapped my head around many, many times how to do this even more specific and precisely. This is extremely challenging, and you have to take into account you need to isolate this input from the contribution of the thalamus and the intracortical feed-forward drive. For example, from layer 4 and layer 6 onto the same compartment. And if you want to, you would need to manipulate and dissect this not only in this pathway, but in the location specific on the dendrite. And that is at least beyond our experimental capabilities.
Thank you, very nice. I was thinking it sounds like you're proposing that with the same axon, we have really like a multiplexing. So one action potential means something, it's one kind of message, but two is a different message, propagates differently, and three is, again, totally different story.
Absolutely. And well, thank you. Then I got some message across, because this is exactly what we're trying to figure out right now. What are these cells telling the downstream targets, right? Because that's been published for a long time. You have this, as I told you, this beautiful somatotropic organization in the first place, and then the first synapse in the cortex, the somatotropy is lost. So these cells at the single cell level can't tell any downstream target which whisker provided my input. They can't. They respond to all the whiskers at the same time. So what we can now show, and there's a beautiful observation, that this kind of broad receptive field of synchronous responses across the population really generates some kind of population code where you essentially can decode, at least in a computer. Let's see whether a neuron can also decode that, but decode nonetheless, despite the receptive fields, which whisker provided the input from any neurons in the barrel cortex. So rather than having one column, one whisker, you know, due to the broadening of receptive fields at the population level, have every whisker in every column in a single spike in a trial by trial basis. And the idea is that this population code essentially informs directly what was the input, no matter what the input was, or the downstream targets, but the temporal profile of these responses provides some kind of additional information to the downstream targets to modulate their activity. And this is something we're currently looking at and trying to do at an experimental level to really isolate the layer 5 cells as a population and to see whether this population code that we would predict in the simulation is really there, but you need to have sub-millisecond resolution and multiple layer 5 cells isolated and identified to be able to do that. Because the challenge here is that intermingling with these layer 5 cells that I'm talking about are other layer 5 cells, which have nothing to do with that, which are very different, which have no bursting mechanisms, or they have bursting mechanisms, but not those bursting mechanisms. They don't like central stimuli per se, but they are intermingling. So to be able to isolate this population, but you really need to isolate the cells first, the cell type first, to be able to measure that. And the only thing I can say is, in terms of sensory information, they can tell everyone what's going on. We also don't see that these mechanisms are reflecting the specific subcortical targets of these cells. We have looked into this. It doesn't matter whether the cell goes to the higher order thalamus, to the pons, to the tectum, or to the brainstem. All of these cells do that. So it seems really kind of a shortcut, so to speak. Sensory information comes in, and the rest of the brain knows what the cortex is now dealing with. That is maybe one of the arguments. Who knows?
Thanks so much. This was really great. But I'm a little confused. So you started out with constructing very detailed networks, and now you stopped with giving us a perspective of information processing, which is completely feed-forward. So what do you think is the whole recurrent connectivity within a particular barrel is doing to it? And the way I understood it, like a single neural perspective, it would suggest that if one neuron bursts, then everybody bursts. So that would suggest a highly synchronous layer 5 burst.
Yes. So no, not every neuron bursts, right? So the probability of these bursts, even in the active touch situation, is still the lowest, right? So in the least number of trials and in the least number of cells, do you see these bursts occurring? So we're talking, and I'm talking about an increase of burstiness, we're talking from sub 1% to about 5% or so, right? So it's not suddenly everyone is crazily bursting simultaneously. So there's still, even when an animal is engaged in some stuff that we did with it, it's not going crazy. So that is a good question. Exactly the number of neurons, their specificity in targeting, and how many bursts do what, and where do they come from, and is there even a way to differentiate between bursts, where we also show that there are even two kinds of triplet burst scenarios where either the pre-stimulus activity directly drives the calcium spike, or where the pre-stimulus just accelerates that the thalamus drives it. So you have these two scenarios, and there are also the timing difference, just a millisecond or sub 2 millisecond difference in these bursts. So there's a lot to be done. Recurrent connectivity, sure, that is an enormous challenge that we are currently facing. How do you, we are focusing here on the feed-forward response.
Yeah,
Not-
it seems too fast to have any recurrence.
There is, of course, recurrence heavily involved. So the period that we're looking at here is about 50 milliseconds post-stimulus, right? So there is, of course, recurrent activity, which we account for, how all the other cells contribute. Of course, I'm simplifying when I'm drawing this. I'm drawing what is necessary, not what is all the other guys which have an influence. So layer 4, layer 2, 3, different interneuron types all have an influence on these responses. That is, if you take them away, something changes, okay? But it's not for the mechanism. And to be able to look at the recurrent dynamics and how, for example, the output from this neuron could impact downstream processing and so forth is one of the challenges that we're currently facing. In terms of the anatomy, we're not so bad yet, right? But there's a lot of higher order connectivity, which we still have to kind of, the possibility space is exploding here to some degree. And on the other hand, there are technical limitations. You can't do that with full compartmental model, right? So one of the challenges that we had, and this was the study I briefly mentioned that we published a month ago or so, is it actually possible to reduce these multi-scale models into some analytically tractable version? And the paper came out as just a little method paper with some beautiful finding, where you can then ask, okay, you have an analytically tractable version of this whole thing, which allows you to do exactly these more population kind of modeling scenarios, where you, however, replace your leaky integrated fire neuron with such a reduced model, right? It's the same complexity, yet it accounts for the dendritic physiology and the spatial temporal distributions of synapses on dendrite. Then you have additional technical challenges because the typical recurrent network don't account for different cell types and so forth. So we're working with Tatiana Chumachenko on that. How can we actually build such population models that consider more than one excited to an inhibitory population and the asymmetries that you have in the connectivity and physiology of those? And this seems to be a challenge in itself. So yeah, step by step.
Okay, we can do a final.
This is very interesting. I was wondering if this can translate into neural rehabilitation when after stroke in the parietotemporal areas, you need to enhance the rewiring of the sensory, not only the motor part of the sensory. Is it better to combine direct electrical stimulation or to enhance relative depolarization as well as combining with direct stimuli instead of only direct stimuli?
Of course, you know, it's always the hope that this very basic research at some point leads to an application, right? But I have to be very frank, this is in the whisker system of the rodent, right? And I showed you how very specific the anatomy, physiology, and so forth of this area is, right? So to be able to simulate how a seizure kind of propagates across the cortex, this is, you can do this for the barrel cortex if you wish. But the idea behind this is that all of this is available, right? From the anatomical models to the data, and we're currently also the whole simulation framework stuff, exactly for that purpose, so that other people which have great ideas of stuff that we can't do and are not interested in could start testing. And there are various people in the world trying to use this for their own questions. Some of them are in development exactly for that, balance excitation inhibition balance and what goes wrong, which may lead to an epileptic seizure in the end. But I would be very careful to claim that this is directly useful for any such application, also in rehabilitation and axon growth.
After a stroke or after a tumor resection, you have to rehabilitate to get like
there's a lot of things one could do, right? And I'm just careful, you know, because this is
really complex for me.
Yes, it is complex, but it's by far not complete. There are many things we don't know, right? And therefore, I would be very careful. These models are sufficient, it seems, to address the questions we are investigating. But as I told you, we spent years to explain these responses, and then we found that the burst cannot be explained. Something's missing in these models, right? So I think it's a good starting point for others to test whether their ideas, maybe on axon regeneration or compensation or who knows what, may or may not work by making lesions here and trying different growth algorithms with different assumptions to recompensate for that somehow. Some people may do that. The idea is take it and do whatever you want with it.
Right, let's